This is an introduction to your genetics notes, and what we're going to do is go through pages 1 through 3 of the practice packet, which is also going to double as your notes. Then we'll do page 7 and then page 13. So we're here on page 1, and this is just going to introduce some of the basic terms. We are going to just kind of start using these terms really quickly. So as soon as you can kind of master them and know what they mean, the better. The sooner the better, because we are going to progress pretty quickly through the unit, which means your foundational vocabulary has to be strong. So let's go ahead and introduce this. I'm not going to talk too much about Gregor Mendel. I know middle school talks about him plenty, but he was the first to get credit for studying a lot of genetics in pea plants. He cultivated and bred pea plants and looked at a variety of traits. And these are some of the traits that he studied, kind of up here on the top of this chart. And he didn't look at them at the same time. He would just look at seed form or just seed color or just pod form and so on. So he would look at only one at a time and notice that certain physical appearances were more prominent than others, and he was the first to get credit for calling things dominant and recessive and so on, so we call him the father of genetics. So just some historical background. We are going to be looking at one trait at a time, at least for the start of the unit, so for this video. Those are called monohybrid crosses. Mono just meaning one trait, and a cross just means to breed two plants or individuals together. All right. So here, where it says name the traits, you can just go ahead and list the traits on the top of this chart. Again, I know this is a long packet. If you can print it, that would be great. But if you can't, then you're just going to kind of have to follow along and jot some of this down on your own sheet of paper. All right, so for some vocab, let's review some vocab. Chromosomes come in pairs. Humans have, you guys should know this, 23 pair for a total of 46. Each pair of chromosomes is said to be, I'm looking for the vocab word here, blank, because they have the same type of blank. So this should be homologous. Each chromosome is homologous because they have the same types of genes. In other words, they have the information to produce the same. We're just going to say traits. We're not going to go ahead and fill that in, just traits. Of the chromosomes in a pair of homologous chromosomes, one came from mom, the mother. The other came from the father. And all diploid organisms have pairs of homologous, we'll just put this in here again, homologous chromosomes. Remember, diploid means you have a set of chromosomes from mom and a set of chromosomes from dad. All right, so here it says homologous chromosomes have genes for the same traits. Humans have 23 pair of homologous chromosomes. And a gene is a section of chromosome DNA. So let's say that this chromosome over here came from mom and this chromosome came from dad. A gene might be a section of that chromosome. So the section here I'm boxing off and kind of circling. Perhaps that's a gene for eye color right there. And again, since both chromosomes are homologous, they will both have the same area of their chromosomes be whatever that gene is. All right. You can have different forms of a gene. So you can have different forms, and those forms are called alleles. There's different versions. So let's go ahead and say, here's our eye color gene just from the previous example. And let's go ahead and just say that mom has brown eyes and dad has blue eyes. So mom's version of the gene gave us a brown copy of that gene, or brown allele, and dad's version of the gene was a blue-eyed allele. And we're going to denote how to, you're not going to have to write the word brown and blue in the future. We're just going to put letters here. So let's keep going. Of the versions, typically one version is going to show up. You're going to be able to look at it and see it. So like for eye color, let's put this back here. Dominant alleles are alleles that are expressed. You can, if you look at the organism, you would see in this case brown eyes. That's because brown eyes is dominant over blue eyes. And we denote a dominant allele by using a capital letter. You can pick any letter you want. I do recommend picking letters that are unique in the way that they look. So for example, if you pick the letter S, this is probably not the best example because a capital S looks the same as a lowercase s, and it's easy to get those confused. So I like to pick letters that look different when they're lowercase and capitalized. So because brown eyes are dominant, and I'm just telling you that for right now. Let's just say mom had brown eyes. We would say that this allele, this version of the eye color gene, is denoted by a capital B. So we'll say capital B equals brown. And blue eyes are recessive, so I'm going to go ahead and, in this example, we'll say dad's eyes were blue. We'll put a lowercase b, and I'll even write the word blue with a lowercase letter here, just so we remember that blue is recessive. Recessive means that in this case, eye color isn't shown in the individual. They might have an allele for blue eyes, but they might not actually have blue eyes when you look at their eyes. All right, if both of your chromosomes, again, we'll stick with eye color here just for an example. If both of your chromosomes have the same type of allele, so either both dominant or both recessive, so let's say mom and dad both had a brown-eyed dominant allele, you would be homozygous dominant. So you can be homozygous dominant. And I'm going to start to abbreviate these words. So homodom just means homozygous dominant, as in this example here. Or you could have, that was mom's, that was dad's. Maybe both parents had the recessive allele. In this case, you are homozygous recessive, homorec, as in this example. Sometimes you will see homozygous individuals referred to as being pure or purebred, or if you're a horse, thoroughbred. That just means both alleles for a given gene are the same, either both dominant or both recessive. So that's homozygous. Heterozygous, you guessed it. Oops, I don't know where my chromosomes went. Let's go ahead and just draw the chromosomes back in here. That was mom's, that was dad's, there's the gene. Heterozygous means you have one dominant and one recessive allele. So let's say maybe mom had brown and dad had blue. So capital B for mom, lowercase b for dad. These are called heterozygous individuals. Another word for that, a synonym, is hybrid, and yet another synonym is carrier. So any of those vocab words describes a heterozygous individual, and yes, you are responsible for knowing all of them. All right. We can refer to the alleles together as a set as a genotype, right? So I know these examples are talking about seeds, but if we're sticking with eye color, I'm going to ignore the chromosomes over here. You have the homozygous dominant phenotype, capital B, the homozygous recessive phenotype, or sorry, genotype, genotype, lowercase b, or you have the heterozygous genotype, one of each. And it doesn't matter which order you put them in. I don't really recommend putting them in the opposite order, although you could. It's just personal preference, but I also think most people like to have the capital letter first. So again, there's nothing wrong with having the lowercase letter first. It's just you'll probably have a preference. All right. So those are the three genotypes that you can have: homozygous dominant, homozygous recessive, and then heterozygous. Remember, genes, genotype. So when they ask you for the genotype, they're asking for the version of the genes. Phenotype, think of pheno, physical. It's physically how that trait manifests. So this would be what you see. So for eye color, capital B would get you brown eyes. Lowercase b would get you blue eyes. And because we have a dominant allele in our heterozygote individual here, capital B, lowercase b would still get you brown eyes because you have at least one capital or dominant allele. So the phenotypes would be brown, two of them would be brown, and then one would be blue. So that's the difference between the genotype and the phenotype. Remember, think of pheno-physical. It's physically what you see in the organism. All right, so during meiosis, when organisms inherit these chromosomes and they inherit these alleles, remember that your cells are, you're going from one parent cell to 4 daughter cells. So here's a set of chromosomes, one from mom, one from dad. And during synthesis phase, we're going to copy these chromosomes. So let's go ahead and label them, and we'll stick with eye color here. So maybe that portion, here's the 2 portions that we talked about earlier. This was a dominant version from mom and a recessive version from dad. So capital for mom and lowercase for dad. And these chromosomes during synthesis, remember the middle part of interphase, are going to copy themselves. We're going to have DNA replication with helicase and DNA polymerase and all that good stuff, the Okazaki fragments, etc. So we're going to make a copy of these chromosomes. Moms and dads. Mom's version right here is a dominant allele, and dad's version right here is a recessive allele. Now, during meiosis, I'm not going to draw all the meiotic steps, but remember you have one parent cell, so here's a parent cell, and it gives rise to 4 daughter cells. 1, 2, 3, 4, and each daughter cell, remember these are connected by a centromere, so we would have 2n equals 2 in our parent cell. In our daughter cells, we're going to have haploids, so these are all going to be n equals 1. So each daughter cell is going to get 1 chromosome, which I'm just going to draw as a line rather than like a little rectangle. And each chromosome gets kind of separated and assorted into a gamete here. So maybe these are sperm cells if this were a male, if this were dad. So maybe this gamete gets that allele and that chromosome. Maybe this gamete gets this allele and that chromosome, so that would be lowercase b. Maybe this third gamete gets this chromosome right here, so that would be a dominant capital B, and maybe this last gamete gets this one. So when you have fertilization, and you don't know necessarily which sperm is going to fertilize the egg, so you could get any of these 4 gametes, therefore you could get any of these 4 chromosomes. So you will only get 1 chromosome from each pair, and therefore only 1 allele from each parent. So let's say that this was the special sperm cell that fertilized the egg, so you would inherit the chromosome with the whose chromosome was this? This was mom's chromosome. Oops, with the dominant version of the allele. Right. So again, we'll talk about what are called Mendel's laws and independent assortment, or the law of segregation of alleles later. But that's kind of the mechanism as to how you would inherit this. Don't forget that I know it's easy to get lost in the letters here, but you do inherit these chromosomes via meiosis. And this is why we've talked about meiosis and fertilization earlier in the year. All right. So can you predict what an offspring will look like? Absolutely, yes, you can. So in this example, let's say that the sperm cell you could inherit two types of sperm, basically. If this were dad's cell up here, the parent cell, you could inherit a sperm cell with a dominant letter or a lowercase letter. So that's what we're going to go ahead and show you here. So either that sperm or this sperm would have fertilized the egg cell. And let's say, oh, you know what? Let's say that mom is heterozygous also, just for this example. So in this example, both parents are heterozygous. How can you predict their offspring? Well, you just basically put these alleles, these gametes, on the side of a Punnett square. That's what this thing is. So if these were from, obviously these would have to be from dad, and these would be from mom. So you can put mom's, I'm not going to draw the egg cells here, but these are basically egg cells on top. Sperm cells can be over here on the left. Again, I'm just drawing the version of the allele. I'm not drawing the actual sperm cell itself, although why not? If you wanted to, you could. Sperm cells here, egg cells here, and then everything in the Punnett square itself is a potential zygote, a possible zygote. Remember, a zygote is a fertilized egg, so you've got 4 possible types of fertilized egg, and what I'm doing here is I'm just sending the letters down and across. So the capital B goes across, the lowercase b goes across, the capital B goes down, and the lowercase b goes down. So there are 4 possible zygotes. And what you're going to notice is that there are some similarities. So what we can do at this point is write what is called the genotype ratio. Or the genotypic ratio. And all we're saying is what are the possible genotypes here? Well, we would say there's 1 capital B, this would be homozygous dominant. There are 2 right here. These 2 are heterozygous. We'd say 2, that, and we'd say heterozygous. And we'd say there's 1. So 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive. So this is the genotypic ratio. This is the full ratio. For the future, you can just write 1 capital here, 2. You can just write what I'm circling. You don't have to actually say homozygous dominant, heterozygous, or homozygous recessive. One thing that you should not do not just write the numbers unless I'm asking just for the numbers. If you wrote 1, 2, 1, you would receive partial credit because you haven't told me that the 1 here is homozygous dominant, that the 2 is heterozygous, and that the 1 here is homozygous recessive. So it would be correct, but it would be incomplete. So just make sure you include the entire genotypic ratio. The last thing here that you need to know is the phenotypic ratio. So this is how many individuals in this case are brown and how many are blue-eyed. Well, here the homozygous dominant and the heterozygous individuals all have brown eyes because they all have at least one dominant allele. Remember, if you have a dominant allele, you're going to have that allele's phenotype. So all of these three are brown. So 3 individuals are brown, and this homozygous recessive individual here would be blue-eyed. So you would say 3 brown to 1 blue, and that would be your phenotypic ratio. All right. Again, the homozygous dominant gets you a brown, the 2 heterozygotes also give you brown eyes, and then that 1 homozygous recessive individual gets you the 1 blue. So basically, if these 2 parents had a child, there's a 75% chance, a 3 out of 4 chance that child would have brown eyes, and a 1 out of 4 chance that child would have blue eyes. All right, we'll do the practice in class. So I'm going to skip all the way down to page 7. And this is just some more practice using Punnett squares. So let's do a quick example together. Let's talk about tall or short in Mendel's pea plants. So in Mendel's pea plants, tall was dominant. So we'll use the letter T for that. So capital T is for tall, lowercase t is for short. And for this example, let's go ahead and say that let's go ahead and let's cross a homozygous dominant individual. I don't know, we can really pick whatever we want. Homozygous dominant, so capital T, and a heterozygous individual. So whenever you see something that says cross, that's all they're asking you to do, is just to write out the parental genotypes, right? So step 2 in this process. Step 3 is to make a Punnett square. So it doesn't matter which parent goes where. Let's put the homozygous dominant parent over here on top. Remember, if this were a male, each of these would be a potential sperm, right? And if this were a female, let's put her over here on the left side. These would be a potential egg that could get fertilized. And then in the middle, in the Punnett square itself, would be all the potential zygotes that could form. So this would be capital lowercase, and capital lowercase. So these are all the possible offspring, all the possible zygotes. So let's do a genotypic ratio. We have 2 capital T. We have 2 capital T lowercase t. So 2 homozygous dominant, 2 heterozygous, and 0 homozygous recessive. And you can simplify this. Anytime you have 0 of something, you can actually just ignore it. And you can also just simplify the ratio here. You can divide by 2 and just say 1 capital T to 1 capital T lowercase t. So 1 homozygous dominant to 1 heterozygous. For the phenotypic ratio, because all of the possible zygotes have a dominant allele, 100% of the offspring are going to be tall. So you would say 4 tall, 0 short. And similarly, if you have 0 of anything, you can just ignore it. The other thing that you can do is, since all of them are tall, feel free to use fractions or percentages, actually. So I would be tempted, this is just my personal preference, to say 100% tall. That's what I would say. But again, you can use fractions or percentages as you are comfortable with. We'll do the rest of these in class. Let's go down to page 13. All right, page 13, and this is the last little portion here, ends with some final vocab that we've been talking about but haven't actually named. So when you have a Punnett square cross and you have the initial parents, that's called the P generation. So for a Punnett square, the P generation would be on the top and the side. That would be the P generation. So let's go ahead and say, let's do A here. So in P's, tall plants are dominant over short plants. Okay. Cross a homozygous tall plant with a short plant. So homozygous tall is capital T, capital T. And if you're short, that means there are no dominant alleles. That means you are homozygous recessive. You have to be. Lowercase t, lowercase t. This is the P generation, the parent generation. So the parent generation is what goes on the sides of your very first Punnett square. All right. When you fill in the Punnett square, just like we've been doing, the individuals inside, the potential offspring, the possible zygotes, that's the F1 generation. It's like the kids. So the results of breeding the parents. These are the genotypes inside the first Punnett square. Again, we've been doing this, we just haven't been calling them the F1 generation. So let's go ahead and say that the genotypic ratio here is 100% heterozygous. Again, I like using the percentages, so that's what I'm going to do. And the phenotypic ratio, they would all be tall because they all have a dominant allele. 100% tall. What is the phenotype of the plants in the F1 generation? They're all tall. All right, let's take two kids and cross them together. So if it's plants, we don't really care. I mean, if these were humans, then yes, this would be incest, but these are plants, so it's kind of, we don't care as much. But yeah, this would be crossing brothers and sisters potentially. So if you cross two F1 individuals, and again, because they're all the same, you can really pick any two. So let's just pick this one and this one. We'll rewrite their genotypes down here. And it says cross them in this Punnett square here. So let's put one of them on top and one of them on the side. And we're going to do the same thing. So these are like the grandkids, right? This is the F2 generation. And let's go ahead and write out these ratios. Genotypic ratio would be 1 to 2 to 1. Again, you could do 25%, 50%, 25%. It's really up to you. The phenotypic ratio, again, these three right here would be tall. And this one here would be short. And then this is basically the same question as that, as the phenotypic ratio. All right. So what you're going to see is a trait that is present in the parents, the P generation. You had a short mom or a short parent plant, kind of skips this F1 generation, doesn't show up at all. It skips this generation here. Remember, all the kids are tall, but then it kind of reappears in the grandchild generation, in the F2 generation down here. So that's the end of the intro notes. This is going to set us up for basically doing everything on our own now. We'll do all of the rest in class. I hope this helped. Remember, be in charge of the vocabulary. Know phenotype, genotype, etc. Dominant, recessive, blah, blah, blah. and be able to do Punnett squares and make all of your generations from P to F1 to F2. All right, so we'll practice this in class, and I will see you there.